The overall goal of the following experiment is to determine the number of neutrophils that have crossed an epithelial cell monolayer in response to infection. This is achieved by collagen-coating transwells before carefully cultivating epithelial cell monolayers using the inverted transwell filter maneuver. As a second step, bacterial pathogen is cultured in order to infect the apical surface of the epithelial monolayer grown on the transwell filters, which causes the epithelial cells to produce endogenous neutrophil chemoattractants. Next, neutrophils isolated from whole blood are added to the basolateral side of infected epithelial monolayers grown on transwell filters for assaying migration of neutrophils from the basolateral to the apical side. Results are obtained that show a significant increase in the number of transepithelial migrating neutrophils observed in response to infected epithelial monolayers based on quantification of myeloperoxidase from migrated neutrophils.
Visual demonstration of this method is critical, as the inverted transwell filter maneuver to grow and infect epithelial barriers is unconventional and therefore can be less approachable for unfamiliar research groups to establish in their laboratory.
The implications of this technique extend toward therapy of airway inflammatory diseases involving infection, such as pneumonia or cystic fibrosis, because novel treatments can be developed and tested to decrease the amount of neutrophil trans-epithelial migration, which may mitigate the deleterious effects of overzealous neutrophilic breach of mucosal barriers.
Along with Mark, who is a clinical fellow in my lab, demonstrating the procedure will be senior technician Waheed Pirzai and postdoctoral fellow Michael Pazos.